Welcome to Biology 102. We are starting off today with our first unit, which is going to be Chapter 11, The Origin and Diversification of Life on Earth. So life on Earth most likely originated from non-living materials. So basically arose from a very different environment than what we experience today. So in the beginning, there was basically nothing. There's going to be things that are very hot. There's going to be rock. There, of course, is going to be molten lava everywhere. There's going to be different gases that are around. But as far as any living type of thing, there's nothing really. So here we are going to go through and give the basic definition of life. Life is going to be defined by two criteria. First, you have to have the ability to replicate, and then you need the presence of some sort of metabolism. So a chemical process by which molecules are going to be acquired and used, and the energy is then going to be transformed to control reactions within that body. So we're going into our first section. We have complex organic molecules that are going to arise from non-living environments. So we have the formation of Earth. It forms around 4.5 to now they're saying about 4.6 billion years ago. It's going to form from clouds of dust and gases that are left over after the formation of the sun. The oldest rocks that we have found here on Earth are going to be about 3.8 billion years old. The earliest life forms are going to be found in these rocks. They are fossilized bacteria-like cells that are going to age anywhere in the range from 3.4 to 3.8 billion years old. So we're going to start to see organisms that are as old as the oldest rocks that we have here on Earth. Biodiversity is also going to start to develop during this time. It's going to give us a variety and variability among genes, species, and ecosystems on this planet. So, the first organisms. There's different theories on this. So theory one, it's going to originate elsewhere in the universe. So they think that life might have traveled here to Earth on a meteor. Little evidence or experimental data, though, is going to be able to support this idea. But this is one thought or one theory. Most scientists are going to believe that life originated on Earth in three distinct phases. So with these phases, we're going to have the formation of small molecules that contain carbon and hydrogen. So the atmosphere is going to include large amounts of carbon dioxide, nitrogen, methane, ammonia, hydrogen, and hydrogen sulfide. These were being produced by the volcanic eruptions that were constantly going on Earth. There's not going to be any oxygen gas present at this time. So the environment's going to be considered basically a warm cradle of life, or, as Darwin puts it, a warm little pond. And that's going to be very critical to the origins of life. So two reasons this is going to be important. Molecules in this environment are going to be able to bond very easily, and the variety of forms and functions are going to be necessary for chemical processes of life here. So theory two is going to be our warm little pond theory. Phase one of this, we have that formation of small molecules that contain carbon and hydrogen. So the conditions on Earth around the time of the origin of life are going to be very different than they are today. In particular, chemical analysis of old rocks are going to reveal that there's no oxygen gas present, like we said, and that the atmosphere is going to include large amounts of those gases that we just talked about. So we see the eruption here in this picture of that early atmosphere. Almost no oxygen gas is present. Small organic molecules are going to eventually form, and these organic molecules are going to provide the building blocks of life. Now, the Urey-Miller experiment is going to be very important. It was conducted in 1953 by two scientists, Urey and Miller. That's where it gets its name. And they're going to demonstrate how complex organic molecules are going to rise from Earth's early environment. And it's going to involve 4 steps. They're going to create a model of this warm little pond that is going to best estimate Earth's early atmosphere. So in there, they're going to have a flask with all of these different things that we just talked about. They're going to subject that flask to their sparks, which are going to simulate lightning. Then they're going to allow it to cool, like the atmosphere would cool over time. So any compounds that were going to be in there that kind of rain back down, that were in that condensation and floating in the air, are going to come back down into the water that's in the flask. And then they're going to wait and wait. And they are going to examine that water to see what happens. So what they're going to discover, that in that flask, they're going to come up with 5 different amino acids that were formed. They reanalyzed this warm little pond in 2008 and find that there are going to be 20 different amino acids present in living organisms that are going to be produced. So we need those 20 different amino acids in order to produce our bodies and other living things. And they found out when they came back to this experiment over 50 years later that all 20 amino acids had eventually formed. Now, lots of times you think it takes a long period of time for things to form. How many thousands of years would it take for these amino acids to form? But in reality, it took very little time, within a week or month, to form the 5 amino acids. And within 55 years, we had all 20 necessary amino acids that had formed by 2008. So this is going to suggest that complex organic compounds could have been produced from inorganic chemicals and lightning energy in the primitive environment of Earth. So that becomes very interesting. And so here's going to be a model of what went on. So we have Stanley Miller Schoen and Harold Urey, which are going to develop simple 4-step experiments that are going to be demonstrated here. So here we have their warm little pond or model of Earth's atmosphere. They're going to heat it. And then they're going to have their condensation, which goes up into the atmosphere. So that creates their pretend atmosphere. They're going to subject it to an electrical charge, which is going to simulate lightning. Then it's going to condense and come back down. And as it cools and comes back down, it goes back into the warm little pond. And over time, that's when they're going to notice that those molecules will form. So what do we get from this experiment? The first demonstration that complex organic molecules could have arisen from Earth's early atmosphere. So now cells and self-replicating systems are going to evolve together to create first life. So more than likely, it's going to originate, they think, from non-living materials. So with this, the second phase in the generation from life to non-life is probably going to be the assembly of the building blocks which make these cell formations. So this is where it's going to get a bit speculative. It's complicated enough to generate a complex organic molecule, but it's going to be a whole lot more complicated to generate an organic molecule that can replicate itself, that can get to the replication phase or stage that researchers believe that enzymes are going to be created. So we're going into what is known as phase 2. Phase 2, we have the formation of those self-replicating, information-containing molecules. So RNA is going to appear on the scene. RNA is going to catalyze reactions necessary for replication. So we can conclude that RNA may have been the precursor to cellular life and is going to lead to the formation of RNA world hypothesis. This is also going to propose the world was going to be filled with RNA-based life before filling it with a DNA-based life. So we think that RNA came about before DNA. Now looking here, this is going to be a fossil that's from 3.4 billion years ago of one of the first living organisms on Earth. So it's been proposed that the early world had self-replicating nucleic acid molecules that are going to carry information on how to replicate and served as machinery to actually carry out replication. So with this fossil that we see here, this one was found in rocks in South Africa and Australia. The cells are going to appear to be prokaryotic, and they're going to be similar to living bacterial cells that we see today. They don't have a nucleus, they have no organelles, and they're going to have circular strands of genetic information. So although superficial, these fossilized cells look like a couple of circles, maybe several lines, it's going to lead to evidence that supports the idea that there indeed are going to be remnants of cells that we can find in these fossil records. So in this, we can include the age of the rocks themselves that have been reliably determined. So we know that they're old, they're 3.4 billion years old. The size of the circles is going to be similar to that of modern day prokaryotes, and the ratio of two carbon isotopes, we have C12 and C13, is going to be more characteristic of fossilized organisms than the typical rocks that don't contain fossils. But many questions are going to still remain, including, were these cells the first living organisms on Earth? And were they descendants of an earlier self-replicating molecule of RNA? Because no earlier fossils have been found yet. So this is going to be a difficult question to answer. So what exactly is life? Well, like we said, it has to have the ability to replicate. So we need self-replicating molecules. We have now seen these start to come about. So they have the ability to carry out metabolism. That's the breaking down of things, the ability to perform chemical reactions within the cell. So from here, that self-replicating mRNA molecule is going to be right on the border. They're going to be able to replicate, but not carry out metabolism. So that needs to be the next step. So then we move on to phase 3, the development of a membrane that's going to enable metabolism and create what we know as the first cells. So they're going to compartmentalize themselves, become self-replicating, and enable numerous aspects of metabolism to occur once they form this cell membrane. So this is going to make it possible for things like chemicals inside the cell to be at higher concentrations than they are outside of the cell, which is going to be very essential for most of the life-supporting reactions. So how did the first cells appear? Were they spontaneous? What happened? So some evidence is going to suggest that the first cells may have simply come together spontaneously. Specifically, researchers have found that mixtures of phospholipids placed in water and salt solutions tend to spontaneously form small spherical units that resemble living cells. So these units may even sprout new buds at their surface and appear to be dividing. Because the cell-like units don't pass any genetic material on, however, they cannot be considered to be alive. At some point, though, in the past, these units incorporated some self-replicating molecules inside. However, maybe by forming around them, such as microspheres, and they may have been important in this third phase of the generation of life from non-life. So the compartmentalization of self-replicating information-containing molecules into what we know today as cells. So if this did occur, the final step in the creation of something from nothing is now complete. All right, so the origins of life, we have it just basically broken down here for you, kind of a recap or a quick review of the 3 phases that we need. So first, we have the formation of small molecules that contain carbon and hydrogen. And so there we are, we see these amino acids coming together to make these things. We have the formation of self-replicating information-containing molecules. Well, here's our RNA that's coming about. And then the development of a membrane. Now we can encapsulate these things. We can encapsulate the RNA and the amino acids. We can change the concentrations from the outside of the cell to the inside. Now they can become self-replicating, they can perform metabolism, and they can enable these cells to be able to undergo different chemical reactions inside. So in summary, the earliest life on Earth, which resemble bacteria, appeared about 3.5, 3.4 billion years ago, not long after Earth was formed. So could have life originated in ice rather than in a warm little pond? Well, this leads us to Theory 3. So with this one, with Theory 3, researchers have been questioning some of the most basic assumptions about life on Earth. So researchers suggesting that this idea had provided something intriguing or some intriguing evidence and proposed some clever ideas to support it. Their ideas build upon a broad consensus about the most important physical and chemical requirements for the initial generation of self-replicating molecules. Conventional assumption is going to be in living cells today, compartments make the duration and closeness of contact possible. But before life appeared on Earth, under warm, wet conditions, some sort of chambers and microspheres may have been spontaneously formed and served this purpose. So the novel approach, it turns out, as water freezes, tiny compartments start to form within ice. On early Earth, low temperatures may have slowed the degradation of any precursor molecules, including RNA, that formed. And the tiny compartments may have held those precursor molecules close together, making it possible for them to react with each other, and thus, possibly, making the first cells. Precursor or requirement 2. So, novel approach here. Although the reactions usually slow down as the temperature drops, some actually speed up. So as water freezes, the ice crystals are going to form from pure water. If there are any impurities present, such as salt or cyanide, they are excluded from the crystals and concentrated in small chambers of liquid within the ice. At the higher concentrations, these molecules are going to collide more frequently, even as the temperatures drop. So as a consequence, certain reactions can occur more rapidly, possibly including the creation and elongation of the first RNA chains. So is it even feasible that ice was present on Earth early as these precursor molecules could have been forming? So researchers have carried out an experiment in which they prepare and then freeze tubes containing seawater and building blocks of RNA. After thawing out tubes, they find that numerous RNA molecules, some long enough to be able to act as enzymes. And recent evaluations of glacier-encased land north of the Arctic Circle suggest that 4 billion years ago, the sun may have been dimmer, and the Earth may have been cooled so much that ice covered the oceans. So some scientists have even gone so far as to describe Earth as a giant snowball at the time. And so sometimes you hear about the giant snowball effect, or giant snowball Earth. So those are our theories of how the first cells came about and how the first RNA or mRNA molecules came about, and even the amino acids that started it all off. Now we're going to head into what's called species. And we're going to talk about how these different species start to divide and start to become biodiverse. So what exactly is a species? Well, a species basically is different kinds of organisms. A cat and a mouse are going to be different things. Cristiano Ronaldo and Beyonce Knowles, on the other hand, are different versions of the same thing. That is, although they are different individuals, they're both humans. So we generally distinguish between different kinds of living organisms, like a rose versus a daisy, or a wasp versus a fly, or a snake maybe versus a frog. Similarly, we're going to lump other organisms into the same group and recognize them as different versions of the same thing. Roses, red roses and yellow roses, Chihuahuas and Dalmatians. So how do we know when to classify two individuals as a member of a different group and when to classify them as two individuals of the same group? So that's going to lead us into our next biological species concept. So with this, we know species are going to be different kinds of organisms. So basically, a species is going to be divided as a natural population of organisms that can, one, interbreed with each other, or could possibly interbreed with each other, and cannot interbreed with organisms outside of their own group. Now, this theory isn't going to be a modern theory. This theory was created around 350 BCE. That came out as the essentialist species concept. So it's the earliest known definition of the term species. So it says all living individuals and inanimate objects sharing and expressing the same essence or internal quality were going to be considered part of the same species. We're going to have people like John Ray in 1686, who said biological species is going to be based on reproductive ability and shared essence. We're going to have Carolus Linnaeus in 1735, who groups of individuals that all share the same morphological features belong in the same species. We'll have A.P. de Candolle in 1813. He says, a group of individuals who share the same morphological features and are capable of reproducing to produce fertile offspring are going to be in the same species. And then we have Ernest Mayr in 1942. He's going to come together with the biological species concept and start to give it some conditions. So he's going to come together and say that it has to have reproductive compatibility. That's the ability of interbreeding individuals in a population to produce fertile offspring, and reproductive isolation. That's the inability of individuals to interbreed and produce fertile offspring with individuals outside of their own population. So this prevents gene flow from occurring, which can lead to speciation. Today, though, many biologists are going to accept the use of the biological species concept to define a species, but its use in defining all species has its limitations. So some of its limitations might be the biological species concept only is going to be useful in defining sexually reproducing organisms, not asexually reproducing organisms. Fossil records don't indicate how the organism reproduced, and it points at which the reproductive isolation occurred. And third, it does not account for hybridization, or when gene flow occurs between two closely related species. So moving on, we have some key features here. We talked about actually interbreeding or could possibly interbreed within their natural populations. So even if they can be produced in captivity, here we see a horse mixed with a zebra. And so here, this is not going to be considered a species, even though they could interbreed and create, because it's not part of its natural population. So we're going to come into what we call some reproductive barriers. So with these reproductive barriers, they're going to influence reproductive isolation by preventing individuals of different species from interbreeding with one another. So two types are going to be based on whether they can reproduce in isolation occurs before or after the formation of the zygote. So we have prezygotic barriers and we have postzygotic barriers. So with our prezygotic barriers, we have courtship rituals, we have physical differences, and we have physical or biochemical factors that involve the gametes. But let's get into this a little bit more. So with a prezygotic reproductive barrier, it's going to contribute to the reproductive isolation before the formation of a zygote by preventing interbreeding or blocking fertilization. So a zygote's that one cell that's created after fertilization. It can exist in a variety of forms. So we got the first form, habitat isolation. So it prevents interbreeding when two species occupy and utilize resources in two different habitats. Examples, we have two closely related species of frogs, the red-legged frog and the yellow-legged frog. They may occupy the same geographic region of central California, but they prefer two different habitats. Red-legged frogs prefer large ponds, while yellow-legged frogs occupy fast-moving streams. Next, we could have what's called temporal isolation. This could occur when two species have different breeding times during the year. Some examples, we have breeding seasons for the red-legged frog, typically occurs in November and March, while the yellow-legged frog typically will breed in seasons that last from April to late June. With the behavioral, we have that occurring in like the courtship rituals of two species that are going to be different. So example of this might be the elaborate behavioral displays of peacocks or intricate, well-choreographed dances that the male blue-footed boobies of the Galapagos Islands do. We have some mechanical isolations. This is going to prevent two species from mating due to differences in their reproductive structures. So for examples, we could see that with this dog and the pig that we see here. And we have gametic isolation. That's going to prevent two species from mating when biochemical differences in their gametes or within the reproductive structures don't permit fertilization. So gametes of, let's say, the giant red urchin and the purple urchin are going to have slightly different surface proteins that make them biochemically incompatible. So next we have our postzygotic barriers. So with these, they occur after fertilization and generally prevent the production of fertile offspring, and often are going to be our hybrids. So they promote reproductive isolation by preventing the development of fertile hybrids of offspring after the zygote forms. So it does perform or make a zygote, but once it turns into a hybrid and it develops, then it can't reproduce. So with hybrid inviability, it's going to prevent the development of viable zygotes during early embryonic cell division. Examples of this would be hybridization of salamander subspecies. These hybrids will die either during embryonic development or shortly after birth. We also have hybrid sterility. That's going to be a second problem. This is going to occur when hybrid offspring are going to be unable to produce viable gametes as adults. So example of this would be hybridization between a male horse and a female donkey, which produces a sterile mule. Or I'm sure you've heard of ligers. The third thing is hybrid breakdown. So this results in sterility of individuals in subsequent generations after viable fertile offspring are produced in the F1 generation. So example of this would be stunted growth and sterility that we observe in cultivated rice plants that are artificially selected over hundreds of years. So how exactly do we name a species? So keeping track of large groups of anything is going to require a lot of organizations. We need an organization system that we can follow. With a huge number of species on Earth, such as the classification system, it's going to be really, really important for us. So biologists are going to use systems developed by Swedish biologist Carolus Linnaeus in the mid-1700s and published his book called Systema Natura, or Systems of Nature. Here's how it works. So for every species is given a scientific name that considers two parts. Genus, which plural is going to be genera or genre, and then you're going to have a specific epithet. So Linnaeus is going to give humans the name Homo sapiens, meaning wise men. Homo is going to be the genus, and sapiens is going to be the specific epithet. The genus is going to be capitalized, and the genus and specific epithet are both going to be italicized. So, for example, the redwood tree, for instance, has the name Sequoia sempervirens. So Sequoia is going to be capitalized, and sempervirens is going to be lowercased, and both of them are going to be italicized. Now, the strength of Linnaeus' system is that it has a hierarchical system. So each element of this system is going to fall under a single element and a level just above it. So in Linnaeus' system, species is going to be the narrowest classification of an organism. The name for every species within the genus is going to be unique, but many different species may be in the same genus. Similarly, many genera are going to be grouped within a family, and many families are going to be grouped within an order. Orders are going to be grouped within a class, and classes are going to be grouped within a phylum. And as Linnaeus originally set it up, the phyla were going to be classified under one of three kingdoms: the animal, the plant, or the mineral kingdom. Now, that's not what we have today, but that's what he created in the 1700s. So today, many of the species classifications that Linnaeus described have been revised, and some of his designations, such as the mineral kingdom, have been left out. Also, all of the kingdoms are now going to be classified under an even higher order of classification, what's called a domain. But Linnaeus' basic hierarchical structure is going to remain, and all of life on Earth is still going to be named using this system, with all organisms belonging to one of the three domains. So looking here, you have the domain bacteria, you have archaea, and you have eukarya. And so everything's going to fall under one of those three all-encompassing domains. So from the top, it's going to be more inclusive. As you go down, it becomes more exclusive, with species being the most exclusive on the list. So like we said, species aren't always going to be easily classified. We're going to have different types of classifications that we're going to need to go by. So biologists, like humans, can be biased. When we investigate this natural world, for example, they often focus on plants and animals, to the exclusion of the rest of Earth's rich biodiversity. So this is going to get them into trouble when it comes to the concept of biological species. Biologist species concept, or the biological species concept, it's going to be remarkable when we use it in describing most plants and animals, but it's going to start to fall short when we try to represent the universal and definitive way of distinguishing many of these life forms that are going to fall under like Eukarya and Archaea. And so here we see some different classification systems. Classifying it as asexual species, we could classify it as a fossil species. We can classify it determining it when that species has changed into another. We can classify it as a ring species, or we can classify it as a hybridizing species. And so we're going to get into these. So the biological species concept is going to define species as a group of interbreeding individuals. But this is going to be useless in distinction when we're talking about asexual species of the world. So remember, we have our single-celled organisms that include things like bacteria or include archaea. We have plants, we have some animals that are going to use this form for reproduction that doesn't involve fertilization or involve two individuals. So the cell or cells of an individual are simply going to start to divide, and that's going to create a new individual. Asexually reproducing organisms don't have a partner or need a partner. They just simply divide. So because asexual reproduction doesn't involve an interbreeding concept, the concept of reproductive isolation is no longer going to be meaningful, and it's going to seem that every individual should be considered a separate species, but clearly that's not going to be a helpful rule to follow. So when classifying fossil species, we have differences in size and shape of the fossil bones from different individuals that can never definitively reveal whether there was a reproductive isolation between those individuals. So this is going to make it impossible to apply the biological species concept here. So it seems reasonable until you consider that your parents, who are the species Homo sapiens, were born to your Homo sapien grandparents, who were born to your Homo sapien great-grandparents, and so on. So if humans evolved from Homo erectus, at what point did Homo erectus turn into Homo sapiens? So it might not be possible to identify an exact point at which this change occurred. So Chihuahuas and Great Danes, generally they can't mate. So does it mean that they are a different species? No, they're still going to be the same species, but they aren't going to be able to mate because of mechanical differences, because they're different sizes. So they will still fall under the same species. So this is another thing. It kind of makes it hard to wrap your mind around what exactly is a species and what isn't. Difficulties in classifying ring species. So we have an example, the insect-eating songbirds called the greenish warblers. So at the center of Asia live some small insect-eating songbirds, these greenish warblers. They're going to be unable to live at higher elevations in the Tibetan mountain range. Because of this limitation, the warblers are going to live in a ring that goes around the mountain. At the southern end of the ring, in northwest India, the warblers are going to interbreed with each other. Then, as the warblers move north along either side of the mountain range, the population is going to start to split. On each side, the warblers are going to interbreed, but warblers from each side that do not interbreed because of the mountain range separating them, these two side populations of warblers are going to meet up again at the other end or the northern end of the ring forest in Siberia, and they can no longer interbreed. So what's exactly going to happen here? Well, we have gradual changes in those warblers on each side of the mountain range that's going to accumulate. So the two populations that meet up in Siberia are going to be sufficiently different physically and behaviorally that they're not going to be able to reproduce, so they become reproductively incompatible. Because the two non-interbreeding populations are connected by gene flow through the populations, there's no exact point at which one species stops and the other begins. So where exactly do you draw the line? The greenish warblers are just one example of more than 20 ring species that have been described. Hybridization. So difficulties in classifying this. Increasingly, we have more and more hybridization. So that's the interbreeding of closely related species that's been observed among plants and animals and all different types of organisms. So the phenomenon is going to fit with the biological species concept as long as post-zygotic barriers have going to evolve so that these hybrids are going to be weak and unable to reproduce. But in some cases, such as butterflies, these hybrids are going to have high survival rates, and they're also going to be fertile. So whether interbreeding with other hybrids or other individuals from either one of the parent species or the other one, it's going to suggest that it's going to border between species that it's not going to be exactly clear cut with these. So most of the time when we think of hybrids, they can't reproduce and create viable offspring. But sometimes they can. So that's the problem with these species. We have morphological species concept, and that's going to be all of these shortcomings are going to have prompted this alternative approach. So these alternatives tend to focus on the aspects of organisms that, other than reproductive isolation defining it, most commonly is going to use what we call the morphological species concept, and that's going to characterize species based on physical features such as body size and shape. So although the choice of which features to use is subjective, an important feature of the morphological species concept is that it can be used effectively to classify asexual species. And because it doesn't require knowledge of whether individuals can actually interbreed, the morphological species concept is going to be a bit easier to use than the biological species concept when it comes to observing organisms in the wild. So how do new species arise? Well, one species is going to split into two distinct species, and this can occur in two distinct phases. We have what's called reproductive isolation. That's going to be where it can isolate individuals of a species either geographically or genetically. And then we have what's called genetic divergence. And there it's going to be accumulation of physical and behavioral differences over time as they adapt to different environments. And so this is going to require more than just evolutionary changes in the population. This would be things such as mutations, genetic drift, and natural selection. So allopatric speciation is going to form a new species when two populations of species are going to be geographically separated from one another and are going to be incapable of interbreeding. So this is going to be a common form of speciation in animals. So here we have a visual, we have initial population, we have something that's going to cause them to be isolated from each other. So we see a stream forming, the stream becomes wider. So they're going to start to change according to what the environment's like on each side of the stream. So looking at these different squirrels, we can see how one of them adapts to its environment, has a more brown color. The other one adapts to its environment on the other side and has more of an orangey red color to it. Another example would be the finches in the Galapagos. They resemble, or they come from the mainland finch, which is in Ecuador. And so you can see how as the islands started to separate and move further and further apart, you can see how they adapted to the food and the environment in their area where they live, and now they can no longer interbreed. Sympatric speciation is going to be the formation of a new species when two populations of species located within the same geographical area genetically isolated from one another and become incapable of interbreeding. Well documented in things like plants through polyploidization. So polyploidization is going to occur when an additional number of chromosomes exist in the gametes as a result of errors during meiosis. And so you could also have autopolyploidy, and that's going to occur when you have non-disjunction during meiosis, and that produces gametes with additional chromosomes as well. And so here you can see an example of this happening with the different species and creating different numbers of chromosomes. So like we said, it is an error during cell division in plants. Chromosomes duplicate, but it doesn't divide properly. And this doubling of the number of sets of chromosomes is referred to as polyploidy. Individuals with 4 sets can no longer interbreed with an individual that only has 2 sets of chromosomes. So self-fertilizing or mating with another individual that has 4 sets can occur. and that gives you an instant reproductive isolation, and now it's considered a new species. So this is where we're going to leave off, and we will pick up with our part 2 of this video in the next video.